The overall goal of this video is to describe the method for sequencing of Psoralen, cross-linked, ligated, and selected hybrids, or SPLASH, in which pairwise in vivo RNA-RNA interactions are mapped in a genome-wide manner.
This technique is a simple, high-throughput method for mapping the RNA interactome in vivo.
This method can help answer key questions in the RNA genomics field, such as how do different regions along a single strand of RNA or between different strands of RNAs interact with each other.
Though this method can provide insights into yeast and human cells, it can also be applied to studies in other modern organisms, including bacteria and mammalian cells.
We first had the idea for this method when we want to come up with a way to map long-range pairwise RNA interaction within an RNA molecule.
Note that the following procedure contains several stopping points. At these times, the experiment can be paused briefly or indefinitely. Details can be found in the accompanying text. Begin this procedure by washing HeLa cells twice with 5 milliliters of PBS. Place the dish vertically for 1 minute to completely drain any excess PBS. Next, add 1 milliliter of PBS containing 200 micromolar biotinylated psoralen and 0.01% digitonin, which increases cell permeability. Take care to add the solution evenly over the surface of the cells. Incubate at 37 degrees Celsius for 5 minutes. Following the incubation, remove the lid of the dish and place it on ice inside a UV cross-linked